Hello friends, today we are going to discuss the membrane lipids. The biological membranes consist of lipid bilayer with associated proteins. The lipid bilayer acts as the selectively permeable barrier for small molecules and ions, while membrane proteins perform variety of functions like receptor, transport and signaling, etc. The relative proportions of proteins and lipids vary from cell to cell. For example, the myelin sheath of medullated neurons is rich in lipids, while the plasma membranes of bacteria, mitochondria, and the chloroplast contains more proteins. The membrane lipids are amphipathic molecules, that is, they consist of hydrophobic tails and the hydrophilic head groups. The hydrophobic interaction between the membrane components with each other and the interaction between hydrophilic head and the aqueous environment results in formation of lipid bilayer. The biological membranes consist of 5 types of lipids, that is Glycerophospholipids, Galactolipids and sulfolipids, Tetraether Lipids of Archaeans, Sphingolipids and the Sterols. The Glycerophospholipids, as the name indicates, are composed of 2 fatty acids esterified with 2 carbon atoms of glycerol, while third carbon of glycerol is bound to the phosphate group. The phospholipids, like Glycerophospholipids and some sphingolipids, consist of phosphate head joined through phosphodiester linkage with the hydrophobic tail. The galactolipids and sulfolipids also have 2 fatty acids esterified to the glycerol, but they lack phosphate attached to the third carbon of glycerol. Glycolipids, on the other hand, consist of simple sugars or more complex oligosaccharides as the polar head. The tetraether lipids of Archaeans consist of very long alkyl chains etherified with glycerol on both ends. The sphingolipids consist of a single fatty acid linked to fatty amine sphingosine. The sterols consist of a rigid system of 4 fused hydrocarbon rings. That means the glycerophospholipids, galactolipids and sulfolipids and Archaean tetraether lipids are glycerol containing membrane lipids, while the sphingolipids use sphingosine, an amino analog of glycerol. First, glycerophospholipids or phosphoglycerides consist of 2 fatty acids esterified with C1 and C2 of glycerol, while third carbon of glycerol form phosphodiester bond with highly polar or charged group. All glycerophospholipids are derived from phosphatidic acid, in which phosphate group is attached to the third carbon of glycerol. The phosphate group is negatively charged at neutral pH. Phospholipids generally have C16 to C18 carbon chain long saturated fatty acids at C1, while C2 has ester linkage with C18 to C20 carbon chain long unsaturated fatty acids. The percentage of saturation determines the fluidity of plasma membrane. That is, for example, the polar bears living in low temperatures have high percentage of unsaturated fatty acids that make membrane more fluid. The double bond present in the unsaturated fatty acids produces kink or bend. As a result, unsaturated fatty acids are not compactly packed, resulting in increase in the fluidity of membrane, which is essential to maintain unhindered flow of essential molecules to the cell. These glycerophospholipids are named according to the polar alcohol attached to the third carbon of glycerol. The phosphatidylcholine consists of choline and are positively charged. The phosphatidylethanol on the other hand, consists of ethanol amine as a polar head, which is also positively charged, The phosphatidylserine consists of serine as a polar head. It is neutral at neutral pH, while the phosphatidylinositol is negatively charged at neutral pH. The ether lipids in some animal tissues like heart, which is rich in plasmalogen, halophilic bacteria, ciliated protists, and certain invertebrates, they contain lipids in which fatty acids are ether-linked with glycerol instead of ester linkage, which is present in Glycerophospholipids. The platelet activating factor released from basophils stimulate platelet aggregation and serotonin release. The second class of membrane lipids includes the galactolipids and sulfolipids. The galactolipids are the most abundant membrane lipids in the biosphere that makes about 70 to 80% of the thylakoid membranes of the chloroplast of vascular plants. Why the plants have evolved galactolipids instead of phospholipids? The answer lies in that the phosphate is a limiting nutrient in most soils which needs to be conserved for critical functions. The sulfolipids present in plant membranes have negatively charged sulfonated glucose head attached to C3 of 1,2-diacyl glycerol. Third class includes Archaean ether lipids, or these are also called as glycerol dialkyl glycerol tetraethers. The Archaea bacteria live in extreme ecological niches like high temperature, which are called as thermophiles, low pH called as acidophiles, high ionic strength called as halophiles, and methane-rich environments called as the methanogens. In order to cope with harsh environments, the membrane lipids of archaeans consist of glycerol ether linked with two very long branched chain fatty acids. The Archaean lipids differ from membrane lipids of other organisms in that they have strong ether linkage, while others like glycerophospholipids, galactolipids, sulfolipids, etc. have ester linkage between the glycerol and the fatty acids. The second point that distinguishes Archaean lipids is that the Archaean fatty acids are very long chain and branched, while other organisms have shorter unbranched fatty acids. The head group consists of second ether-linked glycerol with either phosphate or sugar residues, while other lipids have only a single glycerol molecule attached to two fatty acids. The glycerol present in archaebacterial membranes also differs from the glycerol of other kingdoms in that the central carbon of archaebacterial glycerol is in R configuration, while it is in S configuration in all other kingdoms. Next class of membrane lipids is the sphingolipids. The sphingolipids were discovered by John Tudichem. The human cell membranes contains about 60 different sphingolipids. The sphingolipids are associated with different functions, like they act as a recognition sites on the cell membranes. The human blood groups are determined by the carbohydrate moiety which is attached to the sphingolipids. The sphingolipids differ from other lipids in that they lack glycerol. Instead, they have sphingosine or 4-sphinganine, which is a structural analog of glycerol, and it is a long chain amino alcohol. The sphingolipids also contains only a single molecule of fatty acids, while other lipids like glycerolipids, phospholipids, galactolipids or Archaean lipids have two fatty acids linked to glycerol. The polar head group is attached to the sphingosine through either glycosidic or the phosphodiester linkage. All sphingolipids are derived from a parent molecule called ceramide, which is a structural analog of diacyl glycerol. The sphingolipids are divided into three classes on the basis of their polar head groups as sphingomyelin, neutral or uncharged glycolipids, and the gangliosides. The sphingomyelins, as the name indicates, are especially abundant in the myelin sheath of medullated nerve fibers. Since they consist of phosphatidylcholine or phosphatidylethanolamine as their head group, sphingomyelins are classified as phospholipids along with Glycerophospholipids. Like phosphatidylcholine, sphingomyelins lack net charge on their head group. The second class of the sphingolipids includes Glycosphingolipids. These are also called as neutral glycolipids since they carry no net charge at neutral pH. They lack phosphate but have one or more sugar units linked to C1 of ceramide. The Glycosphingolipids are further classified as the cerebrosides and the globosides. The cerebrosides have a single sugar unit in the form of glucose or galactose. The galactose containing cerebrosides are abundant in the neural tissue, while the glucose containing cerebrosides are abundant in the plasma membrane of non-neural tissues. The globosides, on the other hand, have two or more sugars either in the form of glucose, galactose, or N-acetyl galactosamine as the polar head group. The gangliosides are the sphingolipids that contain complex oligosaccharides as their polar head groups and one or more residues of N-acetyl neuramic acid or the sialic acid. The sialic acid gives negative charge to gangliosides at the neutral pH. The gangliosides are divided into monogangliosides, digangliosides, trigangliosides, etc. on the basis of number of sialic acid residues present in the gangliosides. And the last class of the membrane lipids are the sterols. The sterols consist of a sterol nucleus, which in turn is made up of 4 fused rings. 3 rings have 6 carbon atoms, while the 4th ring is made up of 5 carbon atoms. The sterol nucleus is almost planar and rigid, preventing the free rotation about the carbon-carbon bond. All eukaryotic sterols are derived from simple 5-carbon isoprene subunits. The sterols cannot be synthesized by bacteria but can be exogenously incorporated into the plasma membrane. The eukaryotes, however, have specific sterols. The cholesterol is present in the animals, stigmasterol is present in the plants, and the ergosterol is specific to the fungi. In addition to their role as membrane constituents, sterols perform other roles as sterol nucleus is also present in the steroid hormones that are involved in regulation of gene expression. The bile acids consist of the sterol nucleus and are involved in emulsification of dietary lipids. Since glycerophospholipids, sphingolipids and sterols are virtually insoluble in water, when these lipids were added in water, they form microscopic lipid aggregates. These lipid aggregates reduces hydrophobic interactions and increases the entropy. Like membrane proteins, membrane lipids are also asymmetrically distributed between the two monolayers of a bilayer. For example, phosphatidylcholine and sphingomyelin are present in the outer leaflet, while phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol are abundant in the inner leaflet of the plasma membrane. This asymmetric distribution of membrane lipids, though not absolute, is essential for cell functioning. For example, when phosphatidylserine is present on the outer leaflet instead of inner leaflet, it signals the formation of blood clot by platelets and destruction of cell by programmed cell death. Thank you.